Assalamualaikum warahmatullahi wabarakatuh. Shalom, Om Swastiastu, Namo Buddhaya, Salam Kebajikan. Good afternoon to all of us and welcome back to the next session of today's virtual course 2024 organized by the Faculty of Pharmacy, Universitas Indonesia. Allow me to introduce myself. I'm Rahman Nabila, an undergraduate student of Faculty of Pharmacy, Universitas Indonesia. Ladies and gentlemen, we're now ready to begin the 8th and 9th lectures of the day. The 8th lecture is entitled The Potency of Total Brass Polymers as Drug Delivery Carrier by Dr. Eni Sagita, Master of Pharmacy, PhD from Faculty of Pharmacy, Universitas Indonesia. The 9th lecture is entitled Lipid Nanoparticles for mRNA delivery and strategy for tissue-specific targeting by Dr. Zainal Ahda Islami Mazrat, Master of Science PhD from Monash Institute of Pharmaceutical Science, Australia. Before the lecture starts, I would like to introduce our moderator for the session, Ms. Anissa Lazwardi Larasati, Master of Pharmacy from Universitas Indonesia. Ms. Anissa Lazwardi Larasati, Master of Pharmacy, complete her Bachelor of Pharmacy and Professional Pharmacist degrees at the Faculty of Pharmacy, Universitas Pajajaran in 2019 and 2020, respectively. She got her Master of Pharmacy degree at the Faculty of Pharmacy, Universitas Indonesia in 2024. She worked as a pharmacist at Klinik Bogor Royal Medical Center from 2021 to 2024 and also a pharmacist at Mulia Pajajaran Hospital from 2020 to 2022. Currently, she is a lecturer and researcher at FFUI in the Laboratory of Clinical Pharmacy with research in sport pharmacy and pharmacy epidemiology.
Okay, thank you for the introduction. So good afternoon, everyone. My name is Anissa Lazwardi Larasati, and I will be your moderator for this outgoing lecture entitled The Potency of Bottle Brush Polymers as Drug Delivery Carrier that will be delivered by Dr. Eni Sagita. So before we begin the lecture, please allow me to read the CV. Okay, Eni Sagita is a lecturer in Faculty of Pharmacy, Universitas Indonesia. She obtained her master from Faculty of Pharmacy, Universitas Indonesia on 2014, and PhD from Monash University, Australia in 2021. Before working for Faculty of Pharmacy, Universitas Indonesia, she was a packaging development scientist at PT Dikta Medica. Her research interests include development of dosage form for pediatrics and also cosmetic technology. All right, without further ado, please welcome our honorable lecturer, Dr. Eni. Dr. Eni, the time and screen are yours.
Yeah, is my voice clear?
Yes.
Okay, thank you, Ms. Anissa, for the warm introduction, and also good afternoon, everyone, and thank you for joining this lecture. First, let me share my screen. Okay, you can see my screen right now.
Yes, we can see.
We can see your screen. Thanks.
Thank you. Let me start the lecture. So today I would like to talk about the potency of the battle brush polymers as drug delivery carrier. But before we jump into the complex structure of the battle brush polymer, I would like to give a little bit introduction about polymers for you who are not familiar with it. So the drink bottle that you use and also the button on your shirt, the rice that you eat, and also the paper bag that you use to bring stuff, they're all made of polymers. So polymer itself means many parts. So a polymer is a large molecule made up of many small repeating units, and this small repeating unit is called monomer. So if you have two monomers attached together, we call it dimer. If we have three, we call it trimer. And then as you have more monomers on your polymer chain, you will have oligomer or polymers. And in terms of the structure, you can have a linear polymer like this or something that has branch. And also, you can have, if you have a polymer chain with the same monomers on the chain, you will call it homopolymer. And also, if you have more than one monomer in your polymer chain, you will call it copolymer. This is like the general knowledge about polymer. And another one is that you have to be familiar with the term degree of polymerization or DP, which means the number of monomers in one chain of a polymer. So if I say I have a polymer with DP 500, that means I have 500 monomers in one chain of a polymer. So if you already know about the polymer and you know the function of the polymer in our daily life, I would like to tell you a little bit about the role of polymers as nanocarriers. So in general, we can categorize nanocarrier into three big groups. First is the lipid base, such as liposome or solid lipid nanoparticle, which you might be already familiar with it. Also the metal base, such as gold nanoparticles, silver nanoparticles, and also the nanocarriers, which made from polymer or polymer-based nanocarriers. But if you can see here, even if when you use the lipid-based nanocarrier or metal-based nanocarrier, you will still need polymer to decorate the nanoparticles for stability or for increasing their half-life in the blood circulation. So here we can see that polymer is really essential in terms of nanomedicine world. So what's good about polymer is that you can synthesize nanoparticle with in a more flexible way, and also you can modify it in various kind of architecture, such as copolymer micelles and other structure of polymers, like the battle brush polymers that I would like to talk about on this course. So now, why battle brush polymers? We call it battle brush polymer because the shape looks like the battle brush that we usually use at home. It consists of a backbone at the center, and then there are a lot of side chains attached to the backbone. So the reason why we are interested in the battle brush polymers is because of its unique cylindrical shape, which based on some research, they say if you have nanoparticles that are not round, that has cylindrical shape, you can have a prolonged plasma circulation. It is also less internalized by macrophages, and you can increase the tumor penetration and also the selectivity. And the other reason is because this structure is covalently linked between the side chains and also the backbone. So that means you do not need a assembly process like in micelle or the liposome, which means that it's also more stable in various environments. The other is because it's easy to tailor in terms of the aspect ratio. Aspect ratio is the ratio between the backbone, the length of the backbone, and also the diameter of the polymers. You can also play with the grafting density, whether grafting density is how many side chains that you attach to each monomer in the backbone. So you can have 100% grafting density, 75%, 50%, or 25%. And also we can play around with the composition of the side chain. And if you have a look at this illustration, battle brush polymers have a lot of side chains coming from the backbone. And each side chains can have a lot of functional group, which you can attach to any other small molecules such as drugs, which means that if you want to load the drug into this carrier, you can have more drugs payload compared to the linear polymer, which means it's also perfect for combination therapy. That's when you want to attach more than one drug in one carrier. So you have seen this complex structure, right? So you probably want to know how to synthesize and also how to characterize it. There are three main methods where you can make your battle brush. First is the grafting through, and then we have the grafting to method, and then also the grafting from. I'll go a little bit one by one with this method. So here, first, in the grafting through method, you need a macromonomers. So macromonomer is like a monomer where you can polymerize, but each monomer already have the side chain. So when you try to polymerize this, you will form a battle brush polymers. With the grafting through method, you can characterize it easily, and also you will have 100% grafting density because each unit of the backbone will already have the side chain. The minus of this method is that you will have low conversion of the macromonomers, and also the purification is challenging because you will try to separate two compound with high molecular weight. And the second one is the grafting to method. So the grafting to method, you synthesize the backbone and the side chain separately. And then when you already have both compound, you just attach the side chains into the backbone using a click reaction. With the grafting to method, it's also easy to characterize, and you can have a long backbone. Also, you can have different type of side chain in one brush. The problem with this method is that you probably will have low grafting density because of the steric hindrance when you try to attach the side chain into the backbone. And also the purification is challenging because you have two polymers that you have to separate. And the third one, the last one, is the grafting from. With the grafting from, you normally first synthesize the macro initiator, and then from this macro initiator, this will be a backbone for your brush, and then you can grow the monomer from the macro initiator to make the side chain. With the grafting from method, you can have long backbone and also high grafting density. The purification is also easier because you separate a small molecule from the large molecule compound. And then you can also control the length and the composition of the side chain. But the minus with this method is that you probably will have intra or intermolecular coupling, and also the characterization of the side chain is challenging. So for the characterization, when I synthesize my battle brush polymers, I use these three main instruments to characterize it. You can use any other characterization method if you need it, but this is the main one. First is the gel permeation chromatography, and then the proton nuclear magnetic resonance, or proton NMR, and then you will need also the atomic force microscopy, or AFM. So we use the gel permeation chromatography or GPC to trace the growth of the chain by the increase in molecular weight and also to determine the hydrodynamic molecular weight of the polymers that we synthesize. So the principle of the GPC is basically similar to the HPLC, but instead of the polarity of the molecule, the separation process is based on the molecular weight of the polymers. So here, for example, we have a column filled with beads, porous beads with certain pore size. And then when you have polymers in solution, the polymers will form a coil conformation like this, something like ball. So if you have longer polymers, you will see that you will have a bigger ball compared to the short one. So when you have a mixture of these polymers in a solution and then you pass them through the GPC column, the one that is shorter will make smaller particle, smaller ball. So it will be retained inside the beads porous longer than the bigger one. So when you detect the compound, the polymers using the refractive index detector, you will expect a chromatogram like this. So the bigger polymer, the one with the larger molecular weight, will come up first. So it when you put them in the chromatogram with the retention time as the as the axis, you will see it the peak of the bigger one is coming first. And then we use also the proton NMR to check the monomer conversion and also to determine the theoretical molecular weight of the polymers based on the conversion. So this is what you expect from this analysis. First is when you just, before you start the reaction, the polymerization reaction, you will see the monomer signal like this. And then as the polymerization reaction is going on, you will see that the monomer signals are decreasing, and then you will see the appearance of broad peaks on the other side, which means that your polymers are already formed. The next one is the atomic force microscopy. This is to confirm the shape of the polymers because I would like to synthesize a bottle brush polymer, so I have to make sure that my polymers has the bottle brush shape because we cannot see it from the NMR, we cannot see it from the GPC, so we need an imaging system to confirm the shape. Also, with this imaging, we can use it to measure the dimension of the polymers because I have polymer with 2 dimension, long length and also the diameter. So we cannot use DLS for measuring the particle size. So here is the first step of the synthesis. I'm not going to go into detail of the synthesis process, just the principle. So the first one is I synthesize the backbone. The backbone using this monomers and this little guy named rough agent. So this little guy is used to control the polymerization of these monomers. And the polymerization is confirmed with the NMR, and this is the one that I showed you before. And when I purify the compound, the polymer, I can see here that the peak of the NMR peak of the polymers, and here we cannot see the monomer signal because we already remove it from the mixture. Also from the GPC, this is the chromatogram, not the chromatogram, but it's already processed. So I make a log molecular weight for the axis. And then here you can see that as we go longer from DP50 to 2000, the peak also shift to the higher molecular weight. And to isolate these polymers, I use the precipitation method. So the polymers and the unreacted monomers were reacted in the dioxane water, and they're all soluble in the dioxane water. And then when we pour it into the acetone, as a pore solvent, all the unreacted monomers are still soluble in acetone, but the polymers itself will precipitate because it's not soluble in acetone. And then we just need to the centrifuge it and then remove the supernatant, and then we get the pure polymer. This next step of the synthesis is the synthesis of macromonomers. So from the backbone, we create a macromonomers. If you see the brown dots here, it's the place where the monomers can grow from there to make a side chain. So what we do is we just attach these molecules into the backbone using esterification reaction or conjugation reaction. And we confirm it with the NMR. You see that these two peaks appears after we react. We do this reaction, and also from the molecular weight, it also shift to a higher molecular weight. Means that we already have this molecules attached to the backbone. And then the third step is the grafting of the side chain. So we have the macromonomers. So we just need to polymerize the side chains from these brown dots using these monomers. And then you have the brush polymers. And then also we confirm the polymerization reaction with the NMR. So here for the macromonomers, you have two peaks in here. And then when we grow the side chains, these two peaks disappear and then replace with the broad peaks in here, which correspond to the side chains of the brush polymers. And then we confirm the shape of the polymers that we produce using the AFM. And here we have we synthesize 4 types of brush polymers. First is the short brush, the long brush, and then this is the short brush, but with the grafting density only 50%. And this is the short brush with the grafting density only 25%. This AFM image correspond to each of the brush polymers. If you see here, the short brush polymer showed a quite dense particle, and also the long brush also show a quite dense particle because it both has 100% grafting density, but this one looks longer than this one. When we try to reduce the grafting density, we can also see it in the image here, the 50% and then the 25%, which showed not a cylindrical shape. And unfortunately, the bottle brush polymers that we produce were not soluble in water, so we have to do something with it, like N group removal. So when we try to analyze, we have the tip, this functional group, this group on the tip of the side chains, which cause the compound become not water soluble. So we try to remove these groups and replace it with other groups which are more water soluble, more hydrophilic, such as the carboxylic acid. And the result is that before we replace it, it's not water soluble, but after we replace it with the more hydrophilic group, we have a brush that is more soluble. So next, we want to try the in vitro and in the in vivo studies. We have, for these studies, we have 6 compounds. The main actor is the short brush, and we want to see the impact of the different architecture. So we want to compare between the short brush and also the linear polymer. We also want to see the effect of the different backbone length. So we compare the short brush and long brush, and then we want to see the impact of different grafting density. So we compare between the short brush and also the brush with 50% grafting density and brush with 25% grafting density. And for the additional study, we also try to compare between short brush with the side chain composed of an acryloyl morpholine. And also we compare it with the short brush with the side chain of polyethylene glycol. First, we do the cell toxicity. So the objective is to prove that our bottle brush polymers are not cytotoxic. And then we use MTT assay. You're probably already familiar with the MTT assay, but for you who are not familiar with it, it's actually a colorimetric assay that measures cell metabolic activity and also the growth rate. The principle is that this MTT compound is a water-soluble compound and also have a yellow color. If you have cells that are alive, the cells can metabolize this MTT and convert it into formazine. Formazine is a crystalline compound which is water insoluble and also have a purple color. So what you expect is if your polymers are not toxic, you will see more purple color than a yellow color. So for example, this 96 well plates, this one is yellow color, which means that you have more dead cells because there are not a lot of cells which can metabolize this MTT. And this is the result. We do the cell toxicity to 4 type of cell lines, 3T3, MDA, MB231, HEC 290, and also 41. In general, our breast polymers are not toxic to the cells because we treat the cells with up to 1 milligram per milliliter, and the cell viability is still up above 50%, except for the brush with 25% grafting density in MDI MB231, they show a little bit of toxicity because in concentration of 0.05, they show only 50% cell viability. The next study is the cell association or internalization. So the objective is to know if our bottle brush polymers can be uptake by the cells, can be internalized by the cells. We study it qualitatively by using confocal microscopy and also quantitatively using flow cytometry. And for this study, we label our polymers with Alexa 488, which give green color, green fluorescence color, and we do it using 3T3 cells and also MDA MB231 cells. So this is the result from the confocal microscopy imaging. I'm not going to show you the result, the image from all polymers, but because they all show similar results, so I'm just going to show you the representative images. First is this one is the short brush numb. So remember, so these 4 images coming from the same cells. This one coming from the green channel of the microscope. And if you remember that we labeled the polymer with the Alexa 488, which has green fluorescence. So when we see the green dots here, that means that's our polymers. And then we also have lysotracker. Lysotracker is a fluorescence compound which bind to the lysosome. So if you see the red dots here, that means it's the lysosome. And when and also we have the blue colors here, which correspond to the nucleus of the cells. And when we merge the image from the green channel and red channel and then the blue channel, you will see the yellow dots here. The yellow dot is a color coincidence between the green and then the red. They show, they will result in the yellow color. So that means our polymers are at the same position with the lysosome, which means that our polymers are internalized by the cells. It's inside the cells. And also the long brush showed the same result. Then we try to count how many polymers that are being uptaken by the cells. So we use the flow cytometry, and this is the results. From in general, all polymers show energy dependent endocytosis because in 4 degrees Celsius, you can see almost nothing being uptake by the cells. And when we compare the between the brush and the linear, we can see that the linear show higher uptake than the brush, which probably because of the smaller particle size. But when we compare between the short brush and long brush, they don't show a significant difference. Interesting thing is when we try to compare the short brush and the brush with the short brush with 100% grafting density and with the one that has less grafting density, we can see that as we decrease the grafting density, the cell uptake is increasing. Next, we want to see how our polymers is circulated inside the body. So we do the in vivo pharmacokinetics and the biodistribution study using a polymers that are labeled with a carbon-14 or a radioactive. So what we do is we inject the labeled polymers intravenously using via the jugular vein to the male Sprague-Dawley rats for 24 hours. So we put the two cannulas inside the rats, which are attached to the carotid artery and also the jugular vein of the rats. So the carotid artery is to facilitate the blood withdrawal for the sampling, and the jugular vein is to facilitate the intravenous administration of the polymers. And this is the result. First is we compare between the linear polymer or also the brush. For the brush, they all show similar plasma concentration profile. But interestingly, when we use the linear one, the linear polymer is the plasma concentration of the linear premier is decreasing very quickly, even less than 8 hours, while all the brushes still have high plasma concentration even after 24 hours. And to confirm this, the data is also supported by the detection of the polymers in the urine. First, you can see here the linear num has around like almost 40% of the linear num is found in urine, while the other brushes only a low amount of them is found in urine. And then we also want to know how the polymers are distributed in the organ, especially in the reticuloendothelial system organ like liver and spleen. We can see here that our brush polymers are accumulated in low concentration in the liver, except for the long brush, which show quite a high concentration in the liver, maybe because of its bigger size. And then when we try to compare between the different grafting density, we can see that the plasma concentration is not significantly different. But when we analyze the urine recovery, we can see that as we decrease the grafting density, we can see an increasing amount of the brush being excreted to the urine. Also, in terms of the biodistribution, the three compounds did not show any significant result. And this is the pharmacokinetics parameter of all of the polymers. What I want to highlight is here. When you have the linear num, the half-life is only 2 hours. But when you make it into the brush structure, the half-life is increased around 21 times. It's also correspond to the clearance where for the linear polymer, you have much higher clearance than the short brush here. And then next one, we try to do the in vivo biodistribution in 41 tumor mice models. So before we use the healthy rat, but now we use the tumor mice models because one of the application of the nanocarrier is for the tumor disease. For this study, we used a syngenic model of breast cancer using an immunocompetent mice, biopsy, female, about 5 to 6 weeks of age. And then we used the 41 cells to induce the tumor. And these 41 cells were injected to the fourth mammary gland of the mice. So here you can see the growing tumor around the fourth mammary gland of the mice. And for the polymers, we label them with the Psi 5.5, so we can do the imaging with it. And then we inject it via tail vein. And we use the iFIS imaging system to see where our polymers are inside the mice body. Sorry. So here are the image of the polymers inside the mice body. Here is the blank, which means that we don't give any polymers to this mouse. And then here is the short brush num, long brush, and then also the brush with 25% grafting density. So if you can see here, this is the tumor, and then we can see that it's like our polymers are accumulated in the tumor because we can see it from the iFIS, because we use the 5.5 Psi 5.5 compound, so we can see it from the imaging from outside. And then we also harvest the organ and then try to confirm to ensure that this is not a fake result. So here we have the tumor and then the spleen, pancreas, kidney, liver, heart, lungs. And we can see that the bottle brush polymers are really accumulated in the tumor. And also some of them is on the liver. And here is the quantitative biodistribution in 41 tumor mice models. So it's similar to the rat model, but this one we also use the bottle brush polymer labeled with the radioactive, and then we see here the bottle brush polymers are accumulated in tumor quite high, is similar to the one in the liver and spleen. So the conclusion is all NAM-based bottle brush polymers were relatively non-toxic to the 4 cell lines that we used before, and we can see that it could be internalized by the cells. And also all NAM-based bottle brush polymers exhibited remarkably prolonged plasma circulation with half-life more than 30 hours. It also show low deposition in major reticuloendothelial system organs such as liver and spleen, which is less than 50% except for the long brush, which has 25% accumulation. This result means that our brush polymers show the ability to evade phagocytosis by macrophages. And also the last one, high distribution to the tumor tissue, around 9 to 14% per gram tissue after 24 hours of administration, indicating the ability of our brush polymers to perform passive targeting to tumor tissue. With all these characteristics, we can conclude that the bottle brush polymers can be an excellent candidate for drug delivery application. And thank you very much. I'll give the session to the moderator.
Okay, thank you so much to Dr. Annie for a very impressive presentation and very interesting presentation. So, ladies and gentlemen, let's start the discussion session. Everyone is allowed to ask a question. Please use the raise hand feature. Then you can ask your question directly, or you can write down your question in the Zoom chat box. For anyone who wants to ask question in Bahasa Indonesia, I can help you to translate the question into English. So, anyone want to ask to Dr. Annie? Okay. Actually, I have one question, Dr. Annie, if I can ask. And so, from your topic today. I wanna know what are some promising clinical applications where the bottle brush polymer can be particularly beneficial as drugs delivery system.
Okay, sorry, did you ask about the application of bottle brush polymer for drug delivery system?
Yes,
Okay.
exactly.
Yeah, some research have used the use of the bottle brush polymers for any kind of therapeutic molecules such as small molecule drug and then also DNA, RNA, and then protein drugs. Yeah, there are a lot of application for even the antibody.
Okay, so there are so many applications for this polymer. Okay. Is anyone have a question?
Hi.
Yes.
Oh yeah. Okay. Hello.
Hi,
Okay.
good afternoon. Thank you very much for that wonderful presentation, Dr. Ernie. It's something refreshing for me. I am also in the field of nanocarrier and drug development. And the polymer brushes for me is quite interesting. You showed that polymer science could be used to enhance our drug delivery, right? And from the cytotoxicity results, it just showed that at microgram amount, please correct me if I'm wrong, that the polymer brushes are quite toxic by itself already. Now, laying on that fact, how can we try to use this novel polymer brushes in the formulation of our nanoparticles wherein we want to carry the drug and make it more specific to the site of action that we want. For example, you presented the tumor, but then it also the polymer itself has toxic effects on the liver at microgram amount. So how can we tread on this problem?
Okay, thank you, Mr. John, for the question. Let me share again my
The
PowerPoint.
potency of bottle brush polymer.
Excuse me. Okay. Yeah, so here if we see the trend of the cell toxicity, the data, you can see that actually they are not really toxic to the cells, but except for the bottle brush with 25% grafting density. This might need more investigation why this happened, but in general, the other bottle brush polymers are not really toxic to the cells. This, for example, we use the healthy cells, the 3T3 fibroblast cells, and we can see that even on a high concentration, they don't show, the cells still show more than 50% viability. And also in terms of how to make the delivery of the drugs specific, for example, to the tumor, because here we have, sorry, as I mentioned before, here we have a structure with, sorry, this one. So we have a lot of side chains here, and each side chain can have functional groups. And then with the this abundant amount of functional group, you can attach any small molecules, include the targeting ligand. So with this targeting ligand that you attach, you still have more sites to attach your drugs. And then this targeting ligand can be used to specifically deliver your drugs into the site of action.
Have you tried using this carrier of polymer of yours into a nano formulation? Because thank you for clarifying toxicity. What I saw is that it's above 50%, but for us to say that the formulation itself is biocompatible or the polymer itself is biocompatible, guidelines would denote that it should be above 95% of the cell viability. And moreover, I am more interested about using this modality or this new platform when we try to incorporate it in a nano platform. For example, we would do ionic cross-linking, and the polymer itself already has a hard time going inside the cells if it would have a higher grafting if it would have higher grafting. So how can we maybe we could do some further investigations on this. Definitely an interesting part of your study. So yeah, thank you very much, Dr. Ernie. Wonderful job.
Thank you.
Hello.
Hello.
Okay, thank you for your question. So,
Hello.
okay, Tony.
I would like to ask for some clarity. How are the bottle brush polymer utilizing drug delivery particles?
I'm sorry, can you repeat the questions? It's a bit noisy here, so couldn't really.
I am like, how are the bottle brush polymers utilized in drug delivery nanoparticles? I didn't get you clearly on that.
Okay, so how I use my bottle brush to deliver the drugs? Is that what you mean?
Yes.
Okay.
Yes.
Okay, let me show you again the slides. Maybe, okay, I will try to show you the chemical structure of the bottle brush polymers.
Okay.
Okay, here. So this one is the chemical structure of the brush polymers. This one is the side chains. And these monomers is really reactive. So basically what you need is you just attach any drugs. Okay, this one is carboxylic groups, basically, when this group is released. So basically, if you have with the amide or with the hydroxyl groups, you can attach that easily with these groups. So when you have, for example, drugs with hydroxyl group, this hydroxyl group will attack this group and then will replace this one. And then the drugs will be attached to the bottle brush. And then you will have a drug delivery using the bottle brush as carrier.
Okay. Yes, I've got it now.
Okay, thank you. I think there is
then maybe
okay.
I wanted to just clarify. You explained that the graph density affects the behavior of the bottle brush polymer in nanoparticles. The way I've grabbed it, like the higher the graph density, it increases. There is increase in steric stability and hydrophilicity. I don't know whether I've got it well.
Okay, so the grafting density is for our study, the grafting density is not related to the hydrophilicity of the brushes. The one that influence the hydrophilicity of the brush is the groups that are on the tip of the side chain here, the round dots.
Okay. Okay, thank you very much.
Okay,
Thank
you
you
very
also
much.
for the question.
Okay, there is one question in the chat box. Let me read it. From Fajar Hasmarokarsita, I want to ask whether is it possible to the bottle crush polymer can deliver oligonucleotides with drugs? If can, could this polymer help from degradation from human enzymes such as DNA? Because I see that the polymer itself not shaped to cover trapped inside.
Okay, so I have read some papers using oligonucleotides for using the bottle brush as the vehicle. And yes, you can use it to deliver the oligonucleotides. You just need to change the composition of the side chain so it can cover your oligonucleotides, because normally you need something that is positive charge to bind your oligonucleotides, so you can use it. And also in terms of if you want to just entrap the oligonucleotides into the bottle brush polymers, then don't use the one that has high grafting density, because then you won't have space to wrap it. Just use the one that has 50 or 25% grafting density.
Can I add something on that?
Yeah, sure.
On question.
Sure, Mr.
Yeah, I think for the bottle brush polymer, they have the densely grafted side chain that can increase the drug loading efficiency and provide the controlled release profiles. So, however, there are advantages depend on the specific therapeutic context and the design parameters. That's what I wanted to add. That.
Okay. Sorry, can you repeat the question?
I'm just supplementing on.
All right. Okay.
Like, yeah, on nothing.
Okay, thank you, Mr. Omara.
All right.
Okay, thank you so much for the participant, and many thanks to Dr. Annie for the excellent and insightful presentation and discussion. And I will return the session to the MC. Thank you. Thank you, Dr. Annie.
Thank you, Ms. Anissa and our Honorable Dr. Ernie Sagita for the insightful session. Now we would like to call upon Professor Fadlina Chani Saputri to present a token of appreciation for our wonderful moderator and honorable speaker. To the documentation committee, the floor is yours.
Okay. Thank you very much, Dr. Ernie, for your insightful presentation. It's very amazing. We know the potency of bottle brush polymer as drug delivery carriers. I think it's amazing, and we can do the further research to explore their unique properties and potential application from the bottle brush polymer. So we greatly appreciate your expertise and the valuable knowledge you have shared with us today. Thank you very much.
Thank you, Prof. Fadlina.
Once again, thank you, Ms. Anissa and our Honorable Dr. Ernie Sagita. Ladies and gentlemen, as we move on to the lecture 9, I invite you all to the next Zoom meeting session, of which the link will be provided in the Zoom chat. See you all in lecture 9, which will start very soon.